All right, I'm going to go ahead and get us started. Welcome everybody this morning to the inaugural UCSD Psychiatry Grand Rounds of the Daskalakis era. We are very fortunate, and I'm very happy to welcome our new chair, Dr. Jeff Daskalakis, to our Grand Rounds. As a non-speaker, he's actually been in our Grand Rounds as a speaker before. But Jeff, I want to hand it to you for some welcoming comments.
Thank you, Neil, and welcome, Dr. Jernigan. As I mentioned before in a variety of meetings, it's a real honor and pleasure to lead the department and welcome you all to Grand Rounds as chair. As you all know, over the coming year, we're going to launch an important series of exciting new changes to the department that will help lead us to a new era of clinical innovation, care delivery, systems integration, and translational neuroscience research that will allow us to continue to be leaders in psychiatry and transformative in our field. When I go to the office, I see photos of Dr. Jernigan in the hallway, and since then, I have spoken to several department members about her immense contributions to the department and to the field of neuroscience, and it is a real honor to welcome her back to the department to give Grand Rounds. Thanks again, Neil, for doing such an exemplary job, I have to say, with Grand Rounds, and want to personally thank Dr. Jernigan for delivering the first Grand Rounds presentation to the department under my leadership. Back to you, Neil.
Great. So thank you, Jeff, and welcome, and looking forward to many of these in the future with you. Just a quick housekeeping stuff. Those of you who want CME, CE credits, please remember to scan the QR code at the end of the Grand Rounds. Also, immediately following the Grand Rounds will be our trainee session. All trainees are invited, and early career faculty, just stick around in the Zoom room as the faculty exit. Now to introduce today's Grand Round speaker, we welcome Dr. Tasha Wade, a postdoctoral fellow in the ABCD study, who's working with Drs. Joanna Jacobus and Susan Tapert, as well as with our speakers. So Tasha, take it away.
Thanks, Dr. Swerdlow. I'm really excited for today. I was very excited when I saw the announcement that Dr. Jernigan would be giving this talk. So I wanted to just give you a bit of background on her. So for over 30 years now, Dr. Jernigan has had a productive career as a relentless brain researcher. She has over 220 publications to date, but she first started getting her PhD in clinical psychology at UC Los Angeles and interned, practiced, and conducted postdoctoral research at the Palo Alto VA Medical Center and at Stanford University. She then joined the faculty at UC San Diego in 1984. She is currently professor of cognitive science, psychiatry, and radiology, and director of the Center for Human Development here at UCSD. Her research focuses on neuroimaging of the human brain across the lifespan. And most recently, her work in pediatric neuroimaging has been foundational for the field, first leading to the groundbreaking Pediatric Imaging, Neurocognition, and Genetics, or PINGS study, and now through her work as co-director of the Coordinating Center for the Adolescent Brain Cognitive Development, or ABCD study. As the ABCD study is a centerpiece for her talk today, I'll leave her to tell you all about that. But what I do want to note is that more than being an outstanding researcher dedicated to nuanced and high quality science, Dr. Jernigan is an amazing collaborator and mentor. Her unique expertise in neurodevelopment, neuropsychiatric disorders, and genetics, amongst other areas, combine for a unifying theme of understanding the many factors that give rise to human individual variability, which has really made her a wonderful mentor for numerous students and junior faculty across interdisciplinary fields. She is known for setting aside time for her mentees, dedicating long meetings in her office or the faculty club to guide them. This may be best exemplified by her mentorship of 23 postdoctoral fellows and her serving as the chair of 11 dissertation committees, not to mention the numerous other dissertation committees on which she's been a member. We are incredibly lucky to have her not only as our speaker this morning, but also as a member of our UCSD community. So with that, I'm pleased to introduce today's speaker, Dr. Terry Jernigan, for her Grand Rounds lecture entitled Developmental Population Neuroscience and the ABCD Study. Thanks, Dr. Jernigan.
Thank you very much for that lovely introduction, Tasha. Very generous one, I will say. Okay, so perhaps I should share my screen at this point. Is that?
Yes.
Okay. And, see if I can do this all right. And then. Okay, is that working?
Yeah.
Okay.
It's not sharing just yet. You have to, so exit out of your presenter view, click share screen in Zoom.
Okay. I had done that, but let's go back. Share.
And then show your presenter view.
Okay.
Yep, you're good to go.
I'm good. Okay. Thank you. So good morning, everybody. And thanks again to Tasha, and thanks to everyone for allowing me to come and give a talk to you today. Thanks for the invitation, especially on this very significant occasion. I'm really honored to be able to be the first Grand Round speaker. As many of you know, psychiatry was my home department for, well, let's just say decades. Before I assumed some new roles on the general campus that have made it difficult for me to keep up with my psychiatry family as much as I would like. And I can see that, like many families, this one is continuing to grow, and there are lots of new members whom I hardly know. So I hope this will be an opportunity for me to connect and reconnect with folks here in psychiatry. So what is developmental population neuroscience and this ABCD study? Well, I'm going to tell the story from a personal perspective, starting with the questions that motivate my commitment to this new paradigm. And I think I can say without much risk of controversy that all of us want more people in our societies to thrive intellectually and creatively, and fewer people to become disengaged and perhaps fall prey to damaging dependence on substances or other habits, and for more people simply to achieve a greater sense of well-being. But we would also probably all agree that some people learn more, create more, become more emotionally resilient, and some definitely just have more fun than the rest of us, right? My colleague Anders Dale, for example. But the question really is why? Why is this? And for answers to this question, we could probably achieve a lot less consensus. Is it because people are constitutionally endowed with different personal assets, native intelligence, motivation, optimism, capacity for pleasure? Or is it because of formative, sometimes transformative experiences that either enhance or diminish their lives? Well, for the last 15 years or so, my colleagues and I have been particularly interested in this question, and that is the origins of these kinds of differences between people, which we call behavioral phenotypes. And they can be skills like calculation or literacy, they can be levels of expertise or knowledge, or more ineffable qualities like creativity or emotional or social biases, as we see in personality. In other words, relatively persistent traits and habits, really individuality, if you will. So many people, I'm sure many people on the Zoom here, find questions about the origins of these differences among people intrinsically interesting. But from a biomedical perspective, the incentive to study them comes from the well-documented observation that many seriously adverse outcomes evolve disproportionately in the context of specific behavioral phenotypes. And some of these outcomes are academic disengagement or social dysfunction, but also mood disorders and psychotic disorders, and of course, drug and alcohol dependence. But whatever their origins, one thing is certain, and that's behavioral phenotypes like this do not emerge suddenly at a particular point in time, but more gradually through the cumulative effects of factors influencing them over the entire lifetime of the individual. And these plots of old data from my laboratory show that both calculation skills that are shown here on the left, clearly dependent on instruction and practice, and perhaps more surprisingly, even a basic measure of motor inhibitory function shown here on the right, show a very protracted period of development in children. The x-axis here is between, I think it's 4 or 6, I can't read it myself, and 16. And a central interest of ours is why these 14-year-olds, shown here on the left, have acquired calculation skills more typical of 9-year-olds. And these 9-year-olds have motor inhibitory functions more typical of 15-year-olds. So what do we know that constrains our models of the origins of these kind of differences? Well, we know that they're almost always moderately heritable, but the genetic architecture appears to be highly polygenic. That is, the traits may be influenced by many, many variants in the genome, each exerting only a very small effect. But what are the factors that mediate this genotype to phenotype mapping? And what accounts for the large amount of the variability that cannot be explained, at least statistically, by common genetic variation? So how do we build and test a model of a developing behavioral phenotype? Well, conceptually, at least, we can begin with something like this. We can think of it as, of these phenotypes as a function of, first, the neural genome, which is a bit hypothetical, but we know it exists. And in some cases, effects of environmental factors that directly affect the biology of the brain, for example, on early brain damage or toxic exposures. But much more generally, through the cumulative experiences encountering and interacting with the physical and sociocultural environment of the individual. And of course, through interplay between these factors, things we refer to as gene-environment interactions or gene-environment correlation. Now, how are we going to achieve the kind of integration that we need to improve models of such complex processes? Well, many of us concluded some time ago that we needed to start by observing the developing mind and brain closely and continuously. And here at UCSD, a major first step in this direction was a project called PING, our Pediatric Imaging, Neurocognition, and Genetics, which was funded by the Recovery Act addressing the last Great Recession. And this was a project that was organized with Anders Dale here at UCSD and Natasha Akshamov in our department here in Psychiatry and others here at UCSD and many other leading investigators across 10 other distinguished institutions that are shown here. And the project had only one specific aim, and that was to create a public pediatric imaging genomics database, because one did not exist at that time. There were young adults and there were populations of older individuals, but no pediatric age range imaging genomics databases of any size. And we recruited individuals, participants, or families of participants between 3 and 20. We enrolled over 1,700 of them ultimately, and we had all the key deliverables that were of acceptable quality and over 1,200. The deliverables were very limited in this case. This was a 2-year stimulus package grant, and they included demographics, neuromedical and family history. We did a neurocognitive assessment using the just then released NIH Cognition Toolbox. We added some social emotional phenotypes as part of our participation in another stimulus package project called Phoenix Rising. And then we did a multimodal imaging protocol that included structural, diffusion weighted imaging and some resting state fMRI, and of course, genome-wide genotype. Now, in the interest of time, I'm just going to touch on a few of the interesting observations that we made during using the PING resource. And although the sample was cross-sectional, because of its size, we were able to establish unequivocally that the surface area of the cerebral cortex continues to expand through this early age range until approximately age 10, and then it begins to contract subtly, but continuously thereafter. But we were also able to dig down under this global effect on cortical surface area and estimate the effects at each vertex on the surface of the cortex and create visualizations like these that code for each vertex an age-specific estimate of annualized rate of change. And you can see here that at ages 4 and 10, the cerebral cortex is expanding at every point on the vertex. And you can see from this bar here, I don't know if you can see my cursor or not, but that the yellow areas, for example, at least, are expanding in excess of 3.5% a year. Now, this decelerates a little bit, and you can see over here in this part, in the 8 to 20 age range, that the legend over here has a smaller range. But you can still see that at age 8, the cortex continues to expand everywhere. In this case, the yellow area is in excess of 2% a year. But by age 10, there is tremendous deceleration in this expansion, and it's just barely there. And in fact, there are some parts of the cortex that look a little gray or almost blue. And by age 12, it's clear that there is expansion, shown in the blue values here, which continues gradually, becomes more widespread across the cortex, and actually continues quite long after that, long time after that. Okay. But one thing I want to draw your attention to here, because I'm going to return to it later, is to look at this panel of images at age 10. Just what appears to be the peak of surface area expansion. And clearly throughout this set of images, you get the impression that there's some heterochronicity, that perhaps some parts of the cortex perhaps expand earlier or later, and then begin to decline earlier or later, as though there's some phase difference in different regions in the cortex. But we didn't really have, even with this large sample of 1,100 kids, we didn't really have the precision to show for sure if that was a reliable, those regional differences were really reliable. But I'll come back to that later. But here's another one of my favorite papers that we did from Ping. And I am going to, hopefully I'm going to let my colleague Tim Brown here, who's the first author on this paper, which is on the cover of Current Biology, and asked us to make a little video abstract for it. And Tim does just such a great job of describing the study. I'm going to ask him to tell you about it here, and let's see if I can make this work.
First, we show that structural MRI scans of the brain can be used to predict a child's age with much greater accuracy than has been possible with other types of biological measures. In 885 participants from 3 to 20 years old, using a combination of brain measures from MRI scans, we predicted everyone's age within about a year on average, explaining more than 92% of the variance. What this means is that we've uncovered a developmental clock of sorts within the brain, a biological signature of maturation that accurately reflects age differences regardless of other kinds of differences that exist across people. This essentially means to measure where a person lies along a continuum of biological development made up of periods of different kinds of growth and changes over time, which you might call phases.
So one thing that I particularly liked about this paper is that unlike some data-driven analyses, which this clearly was, this one allowed us to look under the hood a bit to determine which biological signals that we extract from multimodality imaging are most sensitive to chronological age at different points across the age range, which is shown here. It's sort of schematized here in this figure. And this has given us some really interesting hints about the way that maturation cascades through these parameters that we can measure with MRI over time. Okay, so just to summarize very quickly from Ping, Ping has contributed to more powerful data-driven analyses of genetic, imaging, and behavioral measures to model cognitive and brain development. And it's, there have been over 100 publications, certainly over 100 publications that have used the Ping data, and the Ping data resource is now publicly available through the NIMH Data Archive. But the cross-sectional design and the very limited assessments reduce the power of the study to explain, really to explain developing individual differences. And the cohort characteristics in Ping did not closely resemble the population. But Ping certainly helped to lay the groundwork for this longitudinal multi-site study, the ABCD study. And in the wake of Ping and recognizing both the potential of this approach and the limitations, leaders at NIH began to consider the next steps. And at this point, it was well established that neuroimaging reveals robust ongoing biological development of the human brain throughout childhood, through adolescence, and into early adulthood. And work focusing more sharply on adolescence have begun to highlight the degree to which trajectories towards important outcomes diverge during this period. So obviously under the best circumstances, there's tremendous increase in knowledge and creativity and independence, resilience. But we also know that I think I gotta go back there. That it is also the period during which the earliest signs of serious mental disorders often occur. And we know for sure now that there is an uptick in risk-taking during adolescence. Most parents knew that anyway. But it clearly is another serious source of increased risk for young people due to accidents and, of course, experimentation with dangerous substances. Let's go back here. Okay, and we know that health, mental health, social and academic outcomes can be associated with many precursor factors like genetic factors, environmental exposures, and behavioral phenotypes or characteristics. But we also appreciated at that time, we and the leaders at NIH, that more effective interventions to prevent adverse outcomes were going to require better models of the roles and the relative roles of these factors in influencing the outcomes. And it was also better appreciated than before that the limited scope and size, and sometimes the method sensitivity of previous studies had reduced the impact of the research. We'll return to that later too. Okay, so the idea for a large-scale study of the adolescent brain originated with NIDA and was developed within the NIH Collaborative Research on Addiction, or CRAN entity, which is a collaboration between NIDA and IEEE and the National Cancer Institute. But as the planning and the ideas for the study began to unfold, a number of additional institutes and centers became aware of the opportunity that this large-scale study might provide for them to answer other critical questions that were central to their missions. And so at this point, ABCD is supported by 10 institutes and centers of the National Institutes of Health and 4 additional federal partners, the National Science Foundation, the CDC, the National Institute of Justice, and the National Endowment for the Arts, and they're all shown here. And as you can imagine, as additional partners came on board, the aims of ABCD became broader. And at this point, these are the main aims: to develop standards for normal brain development, to measure the individual developmental trajectories across multiple domains and identify factors that may influence them for good or for ill, to look more closely at the relative roles of genetic versus environmental factors and the interplay between these, to study the effects of physical health and activity and sleep on brain development and other outcomes. And of course, with this large cohort, we will be observing the onset and progression of mental disorders. We'll see variability in their course and severity. And we want to understand more about the relationship between mental disorders and substance use. And back to the original impetus for the study, we will be looking closely at how substance exposure affects developmental outcomes and vice versa, how developing attributes of children influence their substance use. And this is the consortium as it is today, with 21 data collection sites at many distinguished institutions around the country. Here at UCSD, we are the host of the Coordinating Center and the Data Analysis and Informatics Center. And something I want to emphasize about ABCD is that this is a large number of, or 23 cooperative agreements, and it is a highly, highly collaborative study with our federal collaborators as well as among ourselves. We have an excellent team of people from the NIH and from some of the other federal agencies as well, who work alongside us every day on our work groups. We have a wonderful project director for ABCD, Gaia Dowling, who's a NIDA employee. And we really work very closely together. And you may be somewhat surprised to find that this works extremely well, that having this close partnership in meeting the challenges of a large, complex study like this and solving problems has been very, very helpful. And this just shows you the national scope of ABCD. You see where our sites are located and our centers. A few things I want to point out about the study. We have achieved a very diverse cohort. We have enrolled 11,880 children, 9 and 10 years of age. And the cohort composition in demographic terms largely matches the targets set for us by our epidemiology consultants who guided us through our recruiting process. We have sort of two cohorts. One is a general population cohort. We're recruited primarily through schools around our sites that were within the catchment area of our sites. And what you see here is this graphic that shows on the inner ring the percentages of each of these groups that were our targets, and in the outer ring, how the actual cohort composition as it is today. And you'll see that we did a pretty good job of matching the rings up and you can see that because the dials are pretty much aligned. And the only discrepancy really here noticeable is that we did under-recruit our Asian participant group, but that we think is partly due to the fact that we have a much greater representation than we were expecting and that the epidemiologists were expecting of people in the other category, which significantly here includes people who report mixed race or mixed ancestry. And so we think this is because it's simply the case that either there are more mixed ancestry young children in the country today, or that people are more aware of their mixed ancestry and they're reporting it more frequently. And so so we have this kind of a lot of people and a lot of those individuals are mixed ancestry with some of these other groups, of course. We also have an embedded twin cohort. These these families were recruited through through birth and twin registry at 4 of our sites that are experienced twin sites, if you will. And they are mostly in the Midwest, and they have a different demographic surrounding them and in their states. And so they are a little bit less diverse, and they struggle a little bit more to achieve the diversity in their sample. But they did a pretty good job, actually. And you can see here that there are 1,730 same-sex twins from birth and twin registry in the sample as well. And it is important to note that there is a lot of relatedness in ABCD in general. These are just rough, just to alert people to the fact that there are over 8,000 children who are single birth kids from the general population. We have 1,730 kids who are same-sex twins recruited at the twin sites. And then, but we have a lot of kids in the study who are full or half siblings to each other, but not same-sex twins, over 1,700 at least. Okay, the main visit schedule for ABCD is that we do a very comprehensive assessment at baseline and biennially after that in 2-year follow-ups, including the multimodal imaging. We do a briefer face-to-face assessment at the odd years, so to speak, and then more frequent phone and web assessments mid-year. I'm not going to go through all the protocols because the protocol is unique at each visit, but just to show you a little bit about the domains of assessment. At every visit, there is both a youth and a parent protocol, and you can find all of the protocols on the ABCD website, abc.org. And it is, every single visit is there with all of the instruments that are and measures that are taken at that time point. This is the baseline youth protocol, where you see that we measure physical health, mental health, neurocognition, cultural and environmental factors. We collect informative biospecimens. We carefully assess substance use. We take some other data sources as well, more passive data sources like actigraphy and things like that. But also, this is our brief summary of our brain imaging protocol, which includes structural and diffusion-weighted imaging, but also functional imaging, resting state, but 3 task fMRI paradigms as well. And those paradigms are monetary incentive delay, a stop signal task, and an emotional in-back task. And this is just a parent protocol, as you can imagine, at this young age. This was 9 and 10 at baseline. We got a lot of information from parents about physical health, mental health, substance use, and cultural environment. And I want to take a couple of minutes to talk about the extremely important contributions of the Data Analysis and Informatics and Resource Center, which the director of which here at UCSD is Anders Dale. We apply an MR acquisition protocol that is referred to as a harmonized human connectome protocol. And Anders and his group have worked closely with the MRI vendors, all 3 of them, to ensure that the protocols are well supported on all 3 vendors, and that the vendors are committed to continuing to support the ABCD protocol over time. Obviously, this is a long-term study. And all of the pipelines for generating the derived imaging measures are produced with fully automated computational workflows. And the DAIRC also manages all the central electronic data capture and the public sharing of raw and derived data from all domains through the NIMH Data Archive. And we also share the computational tools, virtually all the computational tools that are used to produce the data. There are a few important enhancements to the multi-site imaging in ABCD that I'll mention. Real-time motion detection and correction is used for all of the imaging in ABCD, and these methods have been adapted for these advanced multiband protocols that we're applying. And they are used for both the functional and the structural imaging. Novel distortion correction is applied to the diffusion and functional imaging methods to improve the alignment with the anatomical images for better mapping. And we apply a relatively novel multiple B-value, multi-direction restriction spectrum imaging protocol for diffusion, which is really interesting because it allows us to get separate shape-dependent measures of the intra and extracellular components of the diffusion signal. And that's already turning out to be very interesting in development and also in looking at certain effects on the developing brain associated. For example, there was a recent very interesting paper looking at the relationship between BMI and these diffusion parameters in the brains in this young cohort. And of course, another job of the DAIRC is to do cross-vendor, cross-site calibration of imaging data across all these modalities. And I can just tell you, this is the imaging data are vast. It's a vast imaging data set, but I can say that over 20 imaging biomarkers are measured within each structure that you see here in the free surface segmentation, each cortical region that you see here, and each major fiber tract in the brain. And in fact, several different cortical parcellation methods are used so that you can measure your favorite imaging biomarker in each of those regions of interest. Now, another thing that distinguishes the ABCD study, and we're really proud of this, is I think we've really set new standards for a very aggressive open science data sharing model. We send the raw neuroimaging data out the door almost immediately. It's shared through something we call the fast track. But annually, we release the curated data of all of the data that have been acquired essentially since the last release. And we've done 2 annual releases so far. In the last one, the 2.0 release, all of the baseline data in all domains was there, and some early longitudinal data, but not of the comprehensive visits. The 3.0 data release is imminent. It's coming out in the next month or 2. And I'm just going to show you, this is a timeline that you can find on the website again. that sort of summarizes what will be present in each of the data releases. I'm just highlighting this with this red box, the upcoming 3.0 data release, which will include all of a re-analysis or a re-release of all of the baseline data and all of the interim longitudinal data. And of great significance for many people who are interested in imaging, we will for the first time be releasing 2-year imaging data. It's not the entire cohort yet, but it will be over half of the cohort. So this will be the first follow-up with all of the imaging variables and the more comprehensive set of measures overall. Okay, so another very important development of the DAIRC, and this is primarily owing to Wes Thompson, Hauke Bartsch, and now Chun Fan, who have developed what we call the Data Exploration Analysis Portal, or the DEEP for short. This is a cloud-based interactive tool that allows for users to do interactive analyses of the ABCD data using mixed models with many factors pre-coded to facilitate nested multi-level modeling of the data. And it capitalizes on and accounts for the cohort structure, which is very complex, as I've been telling you. And so you have pre-coded variables for controlling for race and ethnicity, if you want, SES variables, site, scanner, and relatedness. It provides model diagnostics and advanced plotting options for exploring the data. It facilitates hypothesis pre-registration if investigators want to do that, something that's becoming more common. It allows expert R scripting to extend the functionality of the standard features, and it gives users the option to save and download an entire self-documented analysis with the R scripts and all the associated model parameters, results, and graphics. And so it's pretty popular for lots of different reasons to explore the ABC and to model the ABCD data. Okay, so you may be wondering, how's the science going? Now, this is a little bit of a feign because I'm not a statistician and I can't tell you much about the statistics, but I will tell you this. This study provides very powerful statistics. And I'm just going to show you a few little highlights of this, just for a taste of what's there. I want to remind you of that in ping, we could see these evidence of sort of what looked like maybe some heterochronicity, maybe some phase differences between different parts of the cortex in terms of when the peak of surface area was acquired in that region and when the contraction began. And you see here in this kind of murky picture at age 10, which showed us the data that we were averaging over between 9 and 11 in the ping study. And we really didn't have the precision to say for sure how reliable those regional differences were. Well, now we have almost 12,000 kids who only vary in age between 9 and 11. That's at the baseline. And so there we can take that additional power and show unequivocally that there is definite real heterochronicity, so that there are definitely some regions shown here in the warmer colors that are continuing to expand in this age range between 9 and 11, at the same time that some regions shown here in the blue colors are beginning to contract. And so this is just one little example of the increased precision that we have with this very large sample size. And there is a lot of very interesting work going on literally all over the world using the ABCD data. 23 countries, users have taken the data from 23 different countries. You know, many, 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 many investigators are doing interesting research all over the world. And I obviously cannot tell you, summarize what's going on out there. However, I am going to highlight some work that I am myself very strongly invested in, and that is a focus of our own ABCD imaging lab here at UCSD. This is just a self-indulgent view of our research, I'll admit. It follows up on an interest that I've been pursuing for a number of years now, and the relationship between individual differences in regionalization of the cortex, that is the pattern of relative surface area in different functional areas, and individual difference variability in cognitive and intellectual phenotypes. The lead investigator on this work in our group is our colleague and ABCD postdoctoral fellow Claire Palmer, pictured here, who presented these prize-winning results earlier this year at the Human Brain Imaging Pacific Rim Conference. And Claire used a novel method for estimating the degree of association between a set of distributed effects, in this case across the cortex, and an outcome variable of interest. And here the pattern of regionalization of the surface of the cortex is related to cognitive and intellectual and intelligence constructs estimated using the NIH Cognition Toolbox. The primary analysis examined the two composite measures estimating fluid and crystallized intelligence, respectively. And we had two questions. The first question was, does this new method provide more power than conventional voxel-wise methods for detecting a relationship between brain and behavioral phenotypes when the effects are expected to be widely distributed in the brain rather than highly focal, as we expected these to be? And secondly, if regionalization patterns could be associated with these two intelligence constructs, do the patterns observed appear to be highly similar, as might be expected if a specific cortical regionalization pattern was a reflection of the classical G factor of intelligence theories, or would they be distinct? So, the findings were that both fluid intelligence and crystallized intelligence are predicted by a pattern of cortical regionalization, and they were predicted better with this multivariable method than by the usual mass univariate approaches. But the patterns associated with these two closely related phenotypes are very distinct, and I realize that you can't see these pictures very well, but this is just this top group, the top panel over here on the right, is just showing you various views of the cortex for the fluid intelligence in the first row, in the leftmost row, and the crystallized intelligence measure in the right, in the middle row. And you can see, I think, just by looking at the pattern of the warm and cool colors, that they are distinct from each other. And this third panel here, the top panel on the right, is really highlighting the distinction, the differences between the pattern. And you can see there are many regions where there's clearly very different effects in different regions. And furthermore, you could even residualize the fluid intelligence measure for the crystallized intelligence measure, there's removing the common variance, and see that it did not change, and that's shown here, it did not change the fluid intelligence pattern at all. So clearly it remains very distinct. And the same was true for residualizing the crystallized intelligence measure for the fluid intelligence measure. It also, which you show down here, also does not change the crystallized intelligence measure. Now, there's a lot of interesting stuff in here, but I'm just going to highlight a few things that we take away from this. But first, let me show you that actually these measures, these composite measures, these estimates of fluid and crystallized intelligence, are constructed by averaging across the original 7 different subgroups of the subtests and the original 7 subtests of the NIH Cognition Toolbox. And for example, the crystallized intelligence composite is really an average of 2 fairly strongly language-related subtests, the reading test and the vocabulary test on the toolbox. And the fluid intelligence composite is essentially an average of the remaining 5 subtests. And these subtests tap a number of different constructs, which we would call cognitive control, executive function, working memory, response inhibition, processing speed, things that are related to the concept of fluid intelligence. But if you look at these patterns, you can see that the 2 crystallized intelligence subtests, the ones that are more strongly language related over here on the left 2 panels, really are fairly similar to each other, as you might expect, given that they're really tapping very similar cognitive systems in the brain, the language system in particular. Whereas the patterns that you see across the different components of the fluid intelligence score are a bit more different from each other, as you might expect, given that they tap different kinds of functions in the brain. And so really you could almost conclude from all of these results that each cognitive phenotype measured in the NIH toolbox is associated with its own distinct pattern of cortical regionalization. So what does this mean? It means that the cortex of the best readers in this cohort of children is not regionalized the same way as the cortex of the best cognitive set shifters, for example. And the best cognitive set shifters, their brains are not quite the same regionalization as even the best encoders of a story sequence. And the take-home message for us is that in this cohort, you could say that different minds of these children are associated with somewhat different brains, at least in terms of the architecture of their cortex. So why are these observations important? Well, they're important to us because they're consistent with a recent model of intellectual phenotypes that sort of challenges the conventional theories that a latent causal factor, what we refer to as G, underlies variation in human intelligence. This new model, called the mutualism model, explains that the psychometric hallmarks of G, as it explains these psychometric phenomena as a consequence of functional interactions between relatively distinct neural processing systems during brain development. Complemented by other work by our group examining the heritability of these phenotypes, the results underscore the importance of determining how learning experiences of children with different neural phenotypes may serve to amplify, or unfortunately in some cases to suppress, these positive interactions. And we think that is the important implication of this different way of thinking about the development of intellectual functions. So let's go back to ABCD. That I want to return to the sort of central focus here, and let's think about what are the limitations of ABCD. What is missing? And a lot of us have known from the beginning that what was missing was the beginning, which is that period in development from conception to age 9, which is missing in ABCD. But help is on the way. NIH has announced its intention to launch another large-scale study, kind of similar to ABCD in some ways, which is going to be beginning in that perinatal period through the neonatal and the early childhood period up to age 9, where ABCD picks up. Also, while the ABCD assessment protocol includes child and parent reports and passive measures of activities and environments and experiences, we are missing the experiences of children in the environments where they spend the majority of their waking hours on most days, and that is in their schools. And that is a serious problem. I always call that the black hole of ABCD, that we were missing really a lot about their experiences in their schools and their educational experiences. And this is something I would really like to see addressed in future initiatives. And for the measurement of many phenotypes, ABCD relies on self-report from parents and youth. We do need more, less intrusive, more robust phenotyping methods, preferably with higher temporal density of sampling in some cases. And as you all know, I'm sure, methodologies like this that are appropriate, I think, even for applying in children, are definitely under development, and we think these will make these approaches even more powerful so that we collect information more in real time and more objectively. And finally, we must remember that ABCD is an entirely observational study, and no matter how good our statistical chops are, we cannot determine causality from observational studies. Okay, and now I'm going to talk a little bit about something we all know about, and that is very relevant to how we should interpret findings coming out of ABCD. This is a slide that I borrowed from Wes Thompson. It's a terrific slide. I really appreciate it. And it talks about replicability, which we all know is of serious concern in our field and in other fields in biomedical research. And replicability is defined here by Wes as effects that are similar if estimated in comparable independent samples. Now, this slide, this graph here, shows the expected distribution of observed rho when the true value is 0.1. That's the blue line there, as a function of sample size. And down here, this is like the log 10 of the sample size. So it goes, you know, from 10 to 10,000 across the bottom, the x-axis there. Now, many small studies down here will fail to detect such a small effect as rho of 0.1. But for variables examined frequently in small studies, published values will emerge and they will be dramatically inflated through a phenomenon we call winner's curse, I guess. Few failures to replicate these inflated effects, nor replications with smaller observed effects, however, will be published just because of the biases in our publishing methods and our conventions. And this means that the neuroimaging literature in particular, for example, is biased towards inflated effect sizes. Now, ABCD size studies, which are shown out here, we should expect to see some major scientific, what I call market corrections or course corrections, because observations from ABCD may force corrections in many research areas so that the effect sizes will be revised downward. And some may be shown to be absent or trivially small. Obviously, this scenario resembles the period in human genetics when GWAS failed to replicate many candidate gene effects. And so we have to resist the despair and initial confusion that was felt by those investigators when they were searching for the genes that endowed heritable traits, and take solace from the rebirth and excitement the field now enjoys, as many of you know, I'm sure, as it gets on with the challenges associated with polygenicity and the complex interplay between genetic variation and environmental factors. So what next? Well, I think taking further cues from human genetics, we need to learn to construct models to describe the additive effects of many factors on the outcomes of interest to us. And this is just a little, you know, like, as I mentioned, I'm not a statistician, but this is just kind of the way I think of it in my head, that we want to be able to model these outcomes and expect that there will be genetic factors, and this includes many genetic variants that will contribute to these outcomes in many cases, and that there will be environmental and experiential factors, again, many of them, some of them correlated with the genotypes, and some of them uncorrelated with the genotypes. And then, of course, there will also be, in some cases, so there could be a significant gene by environment interactions. And so I think we need to move in the direction of expecting our models of the outcomes in order to really account for a significant proportion of the variability to include many factors, because this probably reflects reality. We also need to learn how to and embrace combining resources from across large cohort studies to fill the gaps that we need to fill in order to answer our questions more fully. And we need to move towards testing for causal effects with intervention studies that are based on reliable effects of malleable factors on important outcomes. That is to say, reliable effects in observational studies of malleable factors on our outcomes, because this is a firm foundation for moving toward a real intervention study to try to confirm that there are direct causal effects of those prevention or intervention strategies. So in conclusion, developmental population neuroscience is a new, highly collaborative way to do human developmental science. And it has the advantage that many potentially important factors that may influence the development of the child can be measured in real time. The effects of genetic, environmental, and behavioral factors and their interactions on the important outcomes can be estimated. And hopefully promising targets for new, more personalized interventions to prevent or ameliorate adverse outcomes will emerge. Importantly, I think a major contribution is the open science model, which I think will undoubtedly accelerate progress and increase the impact of our work. So I'm going to stop there and thank everybody for your attention. And I want to thank, of course, all those institutes and federal collaborators that I showed on the earlier slide, all 10 institutes and centers and 4 additional federal agencies, and of course, the entire wonderful ABCD consortium. This is just one shot from one of our recent annual meetings over at the Estancia. And I can tell you on behalf of all of them, I thank you for your attention to ABCD, and I'll be happy to take questions.
Terry, thank you so much for this overview of the project that you've worked with for many years, and is clearly not only providing us with interesting science and pathways forward for understanding the brain and individuality, but also providing a resource for the future, and I think the future of our field. It's a generative process, both in terms of the specific knowledge and what it will hold for us in the future. So I'm gonna open us to some of the questions that are being asked by the audience, and I'd like people to enter your questions in the chat room, and I'll call on you. Let's start with Barb Perry. Barb asks about sex differences in regionalization.
Great question, Barb. Actually, Claire Palmer, whose work I highlighted earlier, is very much interested in this as well, and is beginning to look at it as we speak. We don't have the answer yet, but we're definitely looking at it. I think maybe Claire may be actually on the call, and she may be able to answer for herself, but I think the answer is that there appear to be some, perhaps, but we're still trying to define them and to figure out how best to characterize them. So good question.
I'm going to let Joe Gleason ask his question. Joe, can you unmute your mic?
Sure. Hi, Terry. Great talk. I just was wondering, you mentioned the genetics. I'm wondering what the initial study was done with the GWAS chip, but I'm wondering what the current study and the future studies, are they going to have broader things like whole genome or exome sequencing, and because there's a lot of interest in integrating this kind of data with studies like All of Us or the UK Biobank, where there is a big genetics push. Thanks.
Absolutely. Great. Another great question. There's always the intention that we would do whole genome sequencing. And actually, we've been trying to decide who's going to do it and where and when, and who's going to fund it and all that kind of stuff. And NIH has just announced that they will start to do whole genome sequencing. They're going to roll it out over time. I think they're finding it in pockets of money here and there. Kind of is the impression that I have. And so they are beginning. I think this year they're going to start with about 1,000 participants, and then hopefully continue to. As budgets permit, kind of continue to do it. Of course, it's becoming much more feasible to do it financially, and so we're hoping that there'll be an acceleration in the progress there, and that pretty soon we'll have the whole cohort sequence. One issue is that there's a preference for, at this point, using the blood, and our genotyping was done from saliva. And there's just been this argument within and outside the consortium about whether we would want to use the DNA from the saliva for the whole genome sequencing. And so we have blood samples in a more limited, we're sort of rolling out the blood draws. We didn't want to scare too many kids away in the beginning, and so we've kind of made it optional, and we're rolling it out. Now as the kids get a little older, we're getting much more uptake in the blood samples. NHLBI is now one of our sponsors, and there's a whole panel of blood biomarkers that we're collecting. So if we continue to do the sequencing all with the blood, it'll take a little while for us to get blood samples on the whole cohort. We decided to go and do it with the saliva. We'd have them all tomorrow, but that's another issue.
Terry, I just want to pick up on the next question, which is particularly germane to human studies right now, and that relates to COVID. Erin's question about COVID and what effect will the COVID pandemic have on the longitudinal ABCD study?
Well, as you can imagine, it's having a fairly profound effect on the study since we had a lot of face-to-face visits, and we've been adapting at all of our sites, and our sites, of course, in many different communities, and so the rates of infection are very different from one place to the other, and so on. But we have gone to fully virtual visits for some of our visits, which loses us a little bit of the biospecimens, for example. And we've gone to hybrid visits in many recently at many sites where we do many of the things that can be done virtually with fairly good validity in the virtual session, and then we add a shorter on-site imaging, for example, and a couple of things that are where we require a lot of privacy for the youth, and we don't want them doing it at home. And so just a few things that we think where the validity of them is really critical, that we have them on site. And so we have a very short on-site visit for those critical deliverables that we would otherwise lose because of family. But it's a hodgepodge of whether families want to come in, whether research staff at the sites are ready to return, and so it's been challenging, but we're coping with it, and we hope before long we'll be on a normal schedule. The design, we spent a lot of time talking with our fabulous statisticians like Wes Thompson and his whole biostatistics group, and the design really allows for some flexibility in terms of the exact timing of the visits, because we can kind of extrapolate the developmental trajectories and so on fairly well. And so we're hoping that that we'll still have pretty good coverage of this period. But it's heartbreaking in many ways because the kids are just at puberty now, and we are losing some data that just we just can't have face-to-face visits with the families. They're not comfortable, or we're not open, or whatever. So.
Terry, can I follow up on that? Thank you. I'm curious, in addition to just collecting the data, I guess there's also going to be a question of interpretation. I mean, you've got this huge life experience, the environmental factor, as you mentioned.
Yes, and that was probably part of the other earlier question too. And I neglected to say that we were given funding from several of our sponsors to try to address the impact of COVID on our cohort. And maybe Jeff was thinking about that also. So I'll just say that we rapidly, and really Susan Tapert and her colleagues in the assessment work groups did a magnificent job. Tasha, I think, was extremely helpful in this, in pulling together very rapidly a questionnaire, a survey, to get more information from families about the impact of COVID on their lives and their home situations, their education, and all of that stuff. And we are trying to do it approximately monthly at the moment. So we're trying to track this. Is obviously a moving target. And we are funded so far, I think, to do up to four assessments. It's a optional web-based survey that we're giving the parents and the kids. And so we're not getting absolutely everybody participating, but we are trying to do that. I'm sorry, I cut you off, Ron. Was there anything else you were going to ask?
No, that's what I was getting at. Thank you. Yeah, I mean, it's an interesting natural experiment.
Yeah.
Lisa Eiler asks about how big an effect size is needed, and I'll let her ask her question the way she wants it.
Sure, Terry. Having been involved in Enigma and other big consortia, you know that you have the blessing and the curse of being able to detect these small effect sizes. So I guess I'm wanting to hear your thoughts about when you do find a very powerful and statistically significant small effect in ABCD, how do you follow up on that to understand whether it's meaningful and how it's meaningful? What are some of the steps to understand whether you're finding something that's so small that it doesn't affect anyone's lives, even though it's there, or, you know, that's my question.
Well, that's a big question, Lisa. I'm sure you know, and you know that's there are many parts to that. I mean, I think one of the things about having a very high dimensional study like this is that you can begin to look to see whether these small but highly statistically reliable effects are they mediated by any of the other variables and dimensions that you're measuring in the study. And certainly the extent to which they are relatively independent effects, but are as opposed to being mediated by other effects, it tells you a fair amount. I think if you have that many ways of modeling the effects, of giving you some clues as to what is the underlying nature of them. You know, are they really related to something that would not normally be in the model, but which when it is in the model shows you that it's related to, you know, socioeconomic factors or something like that. So I think it's helpful to have a high dimensional study to look and probe and understand them better. Also, it gives us kind of benchmark. Sometimes what we're finding is that the conventional way that we have related, say, a set of brain imaging phenotypes to an outcome or something like that, or a single brain imaging phenotype, yields a very small effect, but we know that that's a pretty good, accurate estimate of what we get from that phenotype, right? So we can begin to build on that to look at how much is added by a multimodality approach. What kinds of multimodality methods increase that effect size significantly. And so you feel like you're on firmer ground to begin to look to see how we can combine the information that we have about the brain or about the child's experiences or whatever in ways that begin to account for more of the variability, kind of like we do in genetics. You know, how can we begin to account for, with SNP heritability, we can account for more of the variability, but it takes putting a lot of factors in the model, right? And so I think that's kind of every ABCD investigator has their own answer to that question. But I think that's how our group is looking at it. And the exciting thing is that we feel like the results are reliable, and that as we, you know, these estimates they are what they are at this point, and we can look at different versions of them, and the differences between those versions are also meaningful.
Yeah, that's great. I guess the other question following up on that would be, one way to test whether things are meaningful is whether you can change them, like you can improve them. Could you say just a little bit more about any sort of side studies or anything within ABCD that's looking at how changeable things are?
Well, I think that's of course, I like to say it's an observational study. It's an observational study, you know, and of course we have the impression that since it's longitudinal, that we may have opportunity to see where something changes in the child's environment, something changes in the, you know, in the biology of the brain or something like that, and we see an outcome that we think is more plausibly related to that change in the environment or that change in the child's biology. But, you know, ultimately, intervention trials are necessary for, you know, because there are many manifestations, even of genetic factors, that are longitudinal. They roll out of the genome over time. And so you can never be sure that they are not another manifestation of something that was there before. So I think, you know, it is, I think what I was trying to emphasize a little bit in my talk was, I think once you've had a very large-scale observational study and you've looked at some malleable factor that you think is a good candidate for being a target for intervention, and you've really looked at, you know, whether that effect survives kind of covariates that might make it look more plausible as a causal factor than it really is, that is a firmer foundation to go and then do a very targeted intervention study on that factor and find out quickly and efficiently if you can move the outcome with it. And I mean quickly and efficiently in a relative sense, right? As opposed to a lot of studies with, we know how many correlates of our outcomes there are that are very similar to each other and that are highly collinear with each other. And I think, I think the idea here is that you want to make sure that the effect survives trivial mediation by a trivial correlated factor.
I'm going to ask I don't know if Abraham is ready to ask his question, but it's highly relevant to this lecture, and that is that geneticists are now struggling to integrate human and non-human genetic data. And Abraham, I see you on, so I'll pass it over to you.
I didn't mean to steal the mic from you, Jeff. Yeah, so I really like the way you're paralleling what you're doing with the progression of genetics at various points, the transition, for instance, from candidate gene to well-powered studies. And one of the things that now I think the field of human genetics is struggling with is how can it use or integrate data from non-human animals, particularly rodents, but could be primates, could be other species. And I wonder, is there any sense of creating a parallel to ABCD in model organisms, either to ask the experimental questions that you'd like to ask but can't ask in an observational study, or just to collect sort of developmental data to understand how the parallel processes exist in animals?
Well, this is kind of like, my fondest dream, that we would really have well interdigitated studies of the basic neurobiology, especially the basic developmental neurobiology of almost any, especially mammalian species. That would be great. But they were really well coordinated and interdigitated with these studies, in other words, designed to inform each other in some sense. And obviously, the animal studies and neurobiological studies would better inform the human studies. I appreciate that. But and I think that there is an understanding of this and an appreciation for this. And maybe if we had a little better integration between the Brain Initiative and these large human population neuroscience studies, that we could achieve that better. Some people have said to me, well, the human brain maturational process, for example, in adolescence is very protracted. And in the rodent model, adolescence is, people will argue, is it 1 day, is it 3 days, or, I'm exaggerating only slightly here. But so, I think there are challenges, obviously, to making direct comparisons, but I think a lot could be gained by really, working together to say, here are the questions that we have. For example, in imaging, one of the most frustrating things is that we know so little at this point about what really are the cellular underpinnings of these very robust developmental signals that we can measure with imaging now over a very protracted period of time. And it's, we know a lot about embryological changes in cell populations and so on, but we really, in the human brain in particular, the time course of many events that we know are going to happen in the postnatal period, the real time course of them, how this plays out in different cell populations, how late hormonal changes change the architecture of the neuropil or the phenotypes of the neurons or the phenotypes of the glial cells, all that stuff is dark matter to us. We can't really we can't connect the dots because it's just very scant data that allows us to answer questions like that. And I have to think that people, that if we had the right, people around the table kind of designing what is the approach that would best help us to understand the meaning of these signals that we can measure and monitor during life, and what they really are, what's really underlying them at a biological level. That I think we could do a lot better. I just think we've missed opportunities there. And I don't know if this is exactly what you were talking about, but I think that this kind of integration is could be very powerful.
Yeah, no, that's just what I meant. I mean, I think many people at UCSD and psychiatry in particular would probably love to work with you on thinking about what's the right way forward with that, and then maybe seeking separate funding for that. It might be very.
Wow. Well, you a lot of witnesses here who said that. Now we're going to hold you to that for sure. Yeah, I'd love to talk more about that.
That's great.
Terry, a quick question from me here. We've talked about big concepts, cross species, design issues, statistical issues, and I want to ask a small concept or a question. Are there, at this point, I know it's early in the game, but are there anatomical phenotypes that you can identify that account for a significant amount of the risk for developing adolescent pathology and addiction or depression or other things that we treat?
I don't think so. We don't, in ABCD, we don't have, well, first of all, as I explained, I think there are a lot of findings out there. They're highly suggestive, but they're not reliable, and it's not anybody's fault necessarily. It's the way we've been doing the science, right? And so I don't think we know as much as we wish we'd known. We'd known at this point, right? And I think these large-scale studies are going to tell us, give us more accurate views of that. But, our kids are still only, 11, 12, 13 years old. And so some of the outcomes that we are hoping to learn more about how to predict have yet to occur in our sample. So I'd be reluctant to say, oh, well, it's hopeless. We don't even have the outcomes in the sample yet. And the longitudinal data that we think may improve our ability to predict things. But the effects of imaging phenotypes to current individual difference variability in the sample, we're not accounting for a lot of the variability with the imaging measures per se, but it's a complicated answer to that story because with a number of other factors, socioeconomic factors, parental education, things like that, we can actually account for quite a lot of variability, individual difference variability. But those are, again, factors that tend to be related to each other in complex ways. And so this is a challenge. You could look at it from one point of view and say, oh, well, we can sometimes account for 20% of the variability or 25%. That's a lot, actually, out of a set of measures that we have that are all about exposures and environments and cultural factors and the brain. And we can show that there are individual contributions made by these variables that even when they're all in the model, they're needed there to add up to that. And those contributions they may be small, but they're highly reliable. So the model is telling us we can explain a fair amount of variability, and you can generalize those models, and these effects are robust, but we don't yet know how to make sense of those complicated relationships. And that is, I think, more the challenge at this point. And whether in the long run, we'll be able to really account for very much of the outcomes, of the most important outcomes to us from imaging in particular, for example. I think the jury is out on that.
Carla, are you on to ask your question? Carla has a question around there she is.
Hi, yes, I think my question was similar to Abraham Palmer's question, just thinking about other parallel studies that might interdigitate. There's a large group here that's working on the baby brain study, and we've been doing this planning grant to try to plan something. And so I just like to take the opportunity to kind of think about what you've learned in doing this that would be your dream way to combine with these other types of studies, whether it's non-humans or whether it's earlier in phases of development, et cetera.
Right. Well, yes, I mean, here, I mean, the good news is with the baby brain, with this is called the HBCD, the Heal Brain Cognitive Development Study, which, as you mentioned, we're all hoping to be involved in here at UCSD. There's a group here at UCSD involved in that, and it is even, you know, in some ways really the possibility of really connecting up what we do know about brain development from you know, basic studies and so on, with what we see in the immediate postnatal period is better, because we're all, you know, we have a lot of data right up to birth, and then so we can make some connections there. So I think first of all, having the analysis of the data that we're getting about the neonatal brain, which is probably where it's going to start. It's probably for the brain imaging part, going to start in the neonatal period. Be fully informed by good basic developmental neurobiology, and really looking for any opportunity to make contact with that literature and extend it into the postnatal life of human beings will be very helpful. And we're hoping that that will be true, because there are some folks who have been involved in that basic neurobiological research who are you know, very much interested in helping to frame the questions and figure out how best to measure some of the brain biological constructs with imaging. So we are really trying to better inform the approach in looking at neonatal brain development with the basic neurobiology. I mean, I don't know if it's going to work. It's still, you know, challenging, but I think that is what we want to do. I think one thing that's exciting to me, at least, about the HBCD approach and the prospect of having a very strong focus on the neonatal period is that, you know, although unfortunately we can't interview our babies and find out exactly how their minds are developing, or even interview their parents very much about it, we should be able to collect more interesting data with wearables, with new wearable techniques and monitoring techniques that are pretty unobtrusive now. You know, they're in baby shirts and diapers and things, you know, and wristbands and things like that. And we should be able to measure a lot of really interesting behavioral and biomarker data that really may help us to understand more about the infant brain. We'll probably be using, you know, electrophysiological methods more, and I think those will be very interesting in this neonatal period. The challenge, of course, is that, you know, you can image with MRI, you can image a newborn, but pretty quickly you can't image them. They don't sleep through the scanning session, and they move too much, and so then you have a little dark hole in the image in the scanning in the MRI. But we'll be able to fill that in to some extent with EEG methods and maybe some eye movement methods and things like that. So we're still monitoring the developing nervous system from the periphery. So I think, you know, those are the things that I think will be really exciting. And of course, it's going to be very challenging, extraordinarily challenging. My hat is off to you for being a part of that team. That's going to be a really challenging job, but very exciting, just incredibly exciting. And I think, you know, just collecting the information in real time, getting a very good history of the pregnancy if possible, because obviously the prenatal period is extremely important, and reconstructing that in a study like ABCD from the mother's distant memory of what was going on, hardly optimal. Also, I think we're ready to really harvest in a much more robust way the electronic health records, probably, and get more detailed information into the study that way. So lots of opportunities, just really an exciting new development.
Terry, in the interest of time, I want to go quick rapid fire here, and then we'll close it off. But Mariana Turner asks about a bilingual cohort, and Pat Judd asks about whether you're studying maltreatment or interactions between child maltreatment and brain development.
Right. So the bilingual cohort, there are a lot of bilingual kids in ABCD, and actually Maribel Robledo Gonzalez is a postdoctoral fellow who's working with us here at UCSD. She was a graduate student with me in Coxsackie a while ago, and she's been very interested in the bilingual cohort. There has already been one paper published on bilinguality and ABCD by the FIU, a former UCSD guy, Raul Gonzalez, and his group. So there are quite a few. We did take families where the parents spoke only Spanish were included. That's the only group that we included where the parents did not speak English, and that's if they only spoke Spanish. So all of our materials are translated into Spanish. So we actually clearly have a lot more Spanish-English bilinguality than other forms, but we do have some if the parents could speak English well enough to participate in English. But for everything was translated, including all the parent forms, into Spanish.
Great.
So there's mostly that. And then the other question was about, well, we certainly, as everybody knows, maltreatment is a reportable thing, and so we of course do have to report any present and ongoing child maltreatment. But we do ask questions about adverse life events. We ask questions that kind of get at conflict in the family, youth reported or parent reported, violence in the family. So we do have a number of surveys, parent and youth surveys, that try to get at some forms of maltreatment. It's not a very specific focus of the study, I would say, and we've avoided probing too deeply there because of trying to make sure we have good long-term retention of our families and so on. But so I don't know if that's an answer, but I think there'll be some interesting hints there. It may not be the most powerful study of child maltreatment, though.
Jeff?
Terry, thank you for presenting at Rounds today. On behalf of the entire department, it's nice to see such exceptional work coming out of one of our department members. The power and impact of this work, and its as it pertains to the multi-center nature, is something that I think reminds me and highlights the type of work that we can do if we integrate research into our clinics, which is certainly the future of what I hope to accomplish, or one of the challenges, but one of the goals that I hope to accomplish as chair. And I want to thank you for highlighting this at our Rounds today. I appreciate you presenting, and thanks to everyone for attending.
Thank you very much for having me.
Excellent. Thank you, Terry. Thank you, Jeff. I'd like to now invite our trainees to stick around for a special time meeting with Terry as our Grand Round speaker. And here again is the QR code for those who need to scan it. We'll give Terry a minute or two as a break to catch her breath, and then reconvene for the trainee session. Thanks, everybody, and see you next month.